Good morning. Good morning, everyone. Welcome back. So those who was before, I'm glad to see you again. Those who are just new here, so we are continuing here, and you can ask your questions, which I will try to answer as far as I can. But we will start from some previous questions which were already collected before, and some questions I put together in one big sort of theme which I'm talking today. So if you have your questions, just put them in this Q&A section, and then I will see that something is coming up. So I may see what are the questions in between and even interrupt my speech and then answer your questions. And anyway, I will keep enough time to answer as many questions as I can. But you can also send it later. We will try to answer them offline, or maybe answering them next time. Okay, so today our topic is, which was announced. Let me share the screen. Which is a very big topic, and there were quite a number of questions about this. So basically, people are analyzing SNP data, SNPs, and the question is how we can use TRANSFAC to analyze those SNPs, how we can find motifs which are destroyed or created by SNPs, and why should we do that, and what does it bring to us? So what we are losing, what we are gaining when we got the SNPs. So let me first introduce a little bit what we are talking about, and then I will go online and show how our tools can help to deal with the SNPs and the motifs which all together. So let me start my presentation. I think you see it now. Basically, TRANSFAC is about transcription factors. This is our beloved, topic, the transcription factors, how many they are and what they're doing in the cell, so how beautiful they are. So this is actually, we believe that transcription factors are very basic and very important part of the whole organization of life, especially multicellular eukaryotic organisms like us. And transcription factors are really a breakthrough in evolution, which was done some years ago, millions of years ago, when basically there were a number of proteins which are actually acting in the nucleus, and their function is to bind to DNA in specific way. So basically, there are some DNA binding domains, plenty of them, and we're talking about that last time. And these proteins are able to recognize particular patterns, particular motifs, we call them, in DNA and anchor there. And then when they're anchored at that place, then they start doing their job. They activate the genes, they interact with each other, they interact with the whole complex, protein, transfer around the DNA, and they do this gene regulation. And then, of course, we have our genome is variable, and we can have so-called SNPs. SNPs are single nucleotide polymorphisms. So, which means that different people with different organisms, they can have a different, a little bit different DNA. And so some nucleotides are changed. And of course, if it's a big change, which leads to a big change of function, some functions, that's called mutations. But those changes which are bearable by the organism, which can be distributed in the population, so people having them in a lot of variability in our genome, but still we are kind of normal. So we keep those SNPs, but they are not really changing our function too much. And those SNPs can happen in various places in genome. For instance, they can happen in in the regions of genes which encode for proteins, for instance, for transcription factors. And then we have a change of amino acid, for instance, in the transcription factor, and which may change the conformation of that protein, and it will start doing something wrongly or differently. But those changes are really very important, and they often of those changes are really leading to real mutations, change in the function. But most of the SNPs, which are really related to our diseases, in fact, they are linked to our diseases or to our different phenotypes, but they are not touching structure of the proteins, but they are happening in the regions around the genes, basically in intergenic regions, in the regions where the expression of the genes is controlled. So this is so-called regulatory region. And those changes which we observe there, they are sort of more soft. So you can change nucleotide here or there, and it will change the possibility of that factor to bind to that place or not, and that may influence the expression of the respective gene. So if we actually compare it with such analogy, so you can say that if you say that basically there are some proteins which are doing the job, some real workers like enzymes doing some metabolites, and there are transcription factors which are actually giving orders to those workers, like CEOs, like on a nice car, driving. But still, if you have a brake in your car, you go nowhere. So if the car is destroyed by some failure of engine, it will not go anywhere. So that mutations or SNPs in the protein itself and transcription factor, they are very bad, or they cause a lot of changes. But if you consider the DNA, you can consider as a road where those transcription factors are driving. And actually, if you see the gaps in the road, in this place where you have to go, what you can do, you can actually avoid it. You can drive another road, or maybe you can just road the sideway, which means if the factor cannot bind to the place where it should bind or where it usually binds, and it's this place, this site is mutated, well, the factor can bind maybe nearby. And that's we see quite often. The regions which regulate the gene, they are actually often enriched by transcription factor binding sites for a given factor. And so it's more like the quantitative measure. So one site can be destroyed, another is not, and still regulation can proceed. The problem is actually if the whole road is mutated. So which means then you go nowhere. Which means that the whole region, which has to control regulation of certain gene, carries a lot of mutations, a lot of variations, which are not really favor to regulation of that gene. So that means if this region is if a person gets such a region in genome, it can be really damaging and leads to a certain disease. So which means there are not individual SNPs, there are individual SNPs, of course, which may change sites here and there. But what is important, the SNPs which we can measure in our genome, they often mark the whole region in genome, which may carry a lot of mutations and can be important for the regulation of the gene. And if the whole region is transferred to one genome or another through the genetic mechanisms, then you can get this predisposition to a certain disease because of a certain region full of SNPs, full of those changes which are not favorable for regulation of the gene. So like in this case, you see a lot of sites which may be maybe destroyed or created by SNPs, but maybe you can measure just one SNP. You are not really sequencing the whole genome. As you know, usually the GWAS studies, these genome-wide association studies, they are done using some chips, some microarrays, which can measure just the certain SNPs, not the whole sequence of the whole genome. But this SNP can be kind of a marker which marks the whole region, which carries potentially a lot of SNPs, a lot of variability, and can lead to a certain disease. So talking about how that happens, it's actually technically very simple. As you know, in TRANSFAC, for instance, we collect so-called motifs, or we call them positional weight matrices, which are presented here as this logo plot. And these motifs are constructed from known binding sites for a given transcription factor, for instance, NF-κB. And then these logos are used to predict new sites in genome. So if we want to know where NF-κB can bind in genome, we take this matrix from TRANSFAC or from other database, and we apply certain mathematics to scan this matrix through the genome and find those places in genome where it matches the place. So basically, the score of that matrix having higher value than the certain cutoff. But what happens if we have a SNP? So basically, a certain nucleotide, for instance, this G, is changed to a T, to an alternative allele. That means that the score of that binding site in this place drops dramatically, because this G, as you can see, is very important in that place. It's actually very conserved. So, and we can see that the new score of that site is much lower than the existing score. And sometimes it's really changing, and very often changing quite drastically. So from the score, which is really near the maximum, it can drop to absolutely random score if it happens in the very conserved position. It may happen in not very conserved position, then the score is changing not too much. So we can put a cutoff on that delta score. So which means we can see those sites which are changed to very high extent. And in the same way, and as you can see, basically it can be go other way around. For instance, in the reference genome, there was a T here, and respectively there was no NF-κB site predicted, and hopefully also we can predict that this place cannot be a place for binding of a transcription factor NF-κB. But in an alternative allele, we got G, for instance, and that site suddenly appears from nowhere. Just one nucleotide change can create the whole site, because the whole surrounding was kind of predisposed. And then in such a case, we call it a site gain. And in case of mutation which kills the site, we call it a site loss. So then we are talking about the site gains and site losses. This is one way of analyzing how SNPs can influence the sites. But again, in this case, we are talking about the site, the SNP, the change of nucleotide, which actually changing the site by itself. So it either creates the site or destroys the site, right? But what we were talking before is that actually the SNPs are often just markers of a bigger region. So they may not be really responsible for change of the site by themselves, but they marking a region of the genome which may carry a lot of sites of our interest, right? And then if such if we see the variability of that SNP, and we see that this variability is correlating with variability to a certain disease, predisposition to a certain disease, then we may say that this region, the whole region, is important for gene regulation, which is important for that disease, right? So, and to analyze such a concept, we are not just focusing on each position and region which a SNP is occupying by itself, or the sites which are covered by that SNP, but we are taking the bigger region around that SNP, certain nucleotides left and certain nucleotides right, and then we calculate how many sites we can find in that region, and then we have to compare it to something else. And for instance, we can take just random regions of genome, or we can take other SNPs which maybe not correlating with that particular disease, but maybe randomly picked up from the cold SNP database, and then we compare using some simple statistics whether number of sites of a certain type, number of sites for a given factor. For instance, you see here we observe 4 green sites, or let's say 4 sites for NF-kappa B factor, but in a control set of regions, and we call them no sequence set, we don't see those sites at all. So we can say that there is a particular enrichment in that region around the SNP, a given set of SNPs correlated with a certain disease or with a certain phenotype. So then it gives us a hypothesis which transcription factors are important for regulating genes in that particular disease, right? Okay. So, and that's actually can be even observed, in experiments. For instance, you see here you see a region, a bigger region of genome. It's hard to read, but it's here is a MYC gene, and here which is marked by this star, there is one SNP which is known to be correlating with predisposition to colorectal cancer. And it appeared to be this SNP is actually located in some we call them super enhancer, which is occupied, and that's experimental data done. These are a lot of ChIP-seq experiments done in one particular colon cancer cell line for many different transcription factors, and you see these factors are really, binding to that place like bee on the honey. So they are coming to that place, and that SNP is actually marking that place, and stating that this place, this region, is really important for regulating, for regulating genes, respectively the MYC gene, which is a known oncogene, and that's those persons who obtain that region in a certain configuration, they become more predisposed to colon cancer compared to other people who have them have these regions in a different genetic background. Right. Okay. So, and today I want to show one example using our tool. And as an example, I took a set of SNPs which were collected from various, actually using meta-genome analysis, using some genome-wide association studies, but actually comparing different genome-wide association studies for one particular disorder, which is called antisocial personality disorder. Well, in fact, this one particular paper was taken for that particular disorder, antisocial personality disorder, and you can see that they found in that paper several SNPs which are correlating with this disorder, and one SNP which is very interesting, very important, is located in this near this gene, which is in fact non-coding RNA. It's called link 00951. And this, you can see, for instance, on that picture, maybe you are familiar with such diagrams. I see here by different colors, they represent different chromosomes. And here we have a kind of a p-value of each SNP. And then those SNPs which have a very high values of, minus logarithmic p-value, like this one, they are correlating with that particular disorder. And that's that one which was observed here, the SNP which is located near the non-coding RNA. And actually, you can see it's in females and also in males. In males, it's even higher, and actually not really higher, but in comparison to other SNPs, it provides better, higher correlation. And you can also see it in more detail. This SNP, actually, this is this one non-coding RNA, and that's a position of that SNP, and the red means that there is a high correlation and high probability, low probability of having by random chance. So okay, so that's the data, and we want to analyze it using our tools. And the idea is to understand how those SNPs are actually changing there are many SNPs, not just this one, which are correlating with that particular disorder. But we want to understand maybe each individual SNP and also see the whole picture, how those SNPs are changing motifs which may regulate transcription of genes. And then we can see which transcription factors are involved. So for that, let me go to our tool. So for that, we go to our website. So if that's maybe time for you also to look at our website. And here you can register for this tool. It's a pre-registration. Of course, you can try this tool. It's called GeneXplain platform. To do everything what I'm doing today, you need also a license to TRANSFAC database. But to have, first you can do it by pre-registration. So you go here to log in. Here you see this basically geneXplain portal, and I'm log in here as I have already registered in my account, and here we go to the geneXplain platform. So it's logging in. And in the geneXplain platform is it's a view of it. So basically, you see here some topic where you can see the databases which are connected to this user account, the data of the user, the analysis tools, and in the middle of the picture, you see actually different categories which you can start exploring. So you can analyze your data if you have RNA-seq data or ChIP-seq data. And today we are talking about SNPs. So actually these are data about genomic variations. So if you click here, you get a number of categories, and you can load your genomic data, genomic variation data. In terms, actually, we are talking today about SNPs. Results of GWAS studies. So basically, these are tables with SNPs, and each SNP is just an accession number from a DB-SNP database. So it's a list of RS numbers. But these data about genomic variants can be loaded in various different formats. For instance, it can be loaded as VCF file, and today we will not have probably time for this topic, but that's a topic maybe related more towards studying in cancer or in other diseases when you have a full genome sequence of the changes in genome, and then you can by sequencing that genome or various regions of that genome, you get the full picture of the variability. And then those files are usually provided as a VCF file, and then you can load here and also analyze it. Today we are focused on GWAS studies and SNPs, so that's a little bit different topic. Then what we we will talk today is how to identify transcription factor binding sites affected by this genomic variation. And there are a number of pipelines which can be used. And you have here also other tools which can discover once you found those SNPs and genes associated with those SNPs or transcription factors associated with those SNPs. You can further analyze their function and their relation to diseases. You can also go further to analyze the networks where those genes are involved, like signal transduction networks, metabolic networks. This is a little bit of different story. So we may touch that later. But today we are really focusing on the motif, on transcription factor binding sites, and how they are affected by this variability in genome. So in the project section, so you can create your projects, and I have created this one project here where I have already loaded this data from this GWAS study. I can open it. So basically, in that, just for those who are not familiar with the tool, here in this tree, you see a standard tree of projects and folders. And then in each folder, you can have various different tables or files. And the type of the file is actually encoded by this little pictogram. So you see here is kind of table. So it means if you double click on it, it will be open as a table. So you can have other little pictogram like this, which means this is actually a track. Track is a file which gives a position in genome, and then if you double click on it, it will open a different view on that. So I will see it during the presentation. But so far we are looking at this table of the data. You see here there were 709 SNPs which are collected. Well, except the first one, which is just some artifact of collection, or maybe importing of the data actually. So probably it was in the wrong place in that Excel file. But okay, our analysis will just simply skip it. But others are presented by this RS number, which uniquely actually shows the position in genome and uniquely represents which nucleotides are referenced in genome and which nucleotide is alternative. So it's always just a pair. And here you must understand, basically, you may know that already, that there are actually 4 nucleotides, right? Not 2, 4 nucleotides, A, T, C, G, right? And in the reference genome, it might be T, but one SNP is talking about just one change. For instance, T is changing to C. This will be one accession number. If you have in the same position, you have T changing to A, then it will be actually a different accession number. So that's important to remember. So that it's not just a position, it's position plus the nucleotide change. And then important is this P value, which is a P value of that GWAS association. And in that, this list is, in fact, that's already a combined list of several studies, not just that one which I showed, which is this social behavior, social behavior. This list is more combined one, which actually related to anger, to actually aggression behavior, this kind of thing. And when it was a paper where we collected these SNPs, I just loaded them in the genome and in this GeneXplain platform. And you see this first SNP is exactly that one which is near that gene, which is a non-coding gene, link 00951. And the p-value association with the disease is basically 1.6 in 10⁻⁹. It's pretty significant. So we would like to understand that particular SNP, and that's the first thing. And in fact, we want to understand it in the context of all other SNPs. what I mean. Exactly what I was talking in the beginning. So, all those SNPs are in fact correlating to the disease individually, but also all together. So each of that SNP have certain contribution to the disease or to disorder, which means they may work in similar mechanisms. This is actually a certain hypothesis, which is maybe not true. Maybe each SNP is actually acting in its own way. That can happen. But our hypothesis is that there is something common between all these SNPs, and common in two ways. In one way, of course, those SNPs, they influencing the same disorder, which means the downstream from the SNP, as people call it. So which means they affect certain genes, and those genes altogether maybe belong to a certain pathway, maybe several pathways, but maybe biggest pathway is really related to that disease. And maybe one SNP is changing one gene in the phase pathway, another SNP, another gene in the pathway. So they're changing different genes, but the whole pathway is affected here or there or there. And when the whole pathway is affected, then it can lead to that predisposition to that disorder. But what is our interest is actually is a little bit a different direction. We are talking about upstream. What I mean. So all these SNPs are changing some motifs in genome. And there is a hypothesis that maybe many of them are changing similar motifs. And then basically, for instance, if many of them are changing motif for NF-kappa B, then generally we can see that NF-kappa B transcription factor is involved in regulation of those genes. And that's a bit looking more upstream. And why we're looking upstream, that's another story. That actually leads us to a question how we can control all this thing together, because upstream means something from upstream regulator. So, if the same similar transcription factor leading to, actually regulating those SNPs or those regions where SNPs are observed, then we somehow can influence on those regulators, maybe with some drugs, and then we can influence to the disease, right? Although when we talk about downstream, we may be too late. So, okay, these genes are already changing and the pathways are changing, but how to influence the whole pathway? It's very difficult. Rather, maybe we can influence on some few master regulators. We can change the whole story. That's why this upstream analysis is very important for us. Okay, a lot of words, but let me just show a bit more on the slide or on really online. Okay, so we have this table. So then we go to the start page, and we will start this pipeline, which is called Analyze SNP List using TRANSFAC. Very simple. So what does it do? Actually, here's this pipeline. It has a certain steps, quite number of steps. And just for, if you are new here, so what we see here, each of these blue boxes is one program or one method, which are connected to each other by intermediate files. So, and these pipelines can be built by yourself. There's when you click on edit workflow, you can change this pipeline, you can add new functions, new tools to it, or you can create the full one from scratch by just adding drag and drop methods from this method analysis section. I will go there a bit later. But now this pipeline is already ready. So for we made it before. By the way, in the tool we have several hundred, well, more than 100 different pipelines, and which are already ready for you to do, and then they can to work, and they can do various things. So when the pipeline is ready, then what you do, you just take your list of SNPs and drag and drop it here. Just another short words about the table itself. Actually, for the pipeline to work, the only thing you need is just the first column. So you need just a list of SNPs. All other columns are for you to have, but you know, in your hands to do some filtering maybe. But basically, for the analysis, we just used here in this particular case just a list of SNPs. We consider them sort of equivalent to each other, which is not necessarily the case, and that we may talk about that a bit later. But now it's they all considered sort of equivalent. Sort of all of them are important for the regulation or to of the genes connected to that disease. And then one parameter of that pipeline is which distance we have to consider around the genes. Why genes? Basically, this is just part of that pipeline, but it could be not necessarily. You can analyze all the SNPs independently from the gene location. But here in this particular pipeline, we consider the most important SNPs are those which are located either in the body of the gene or somehow nearby. So here the default is 10 kb upstream or downstream from the end of the gene or upstream from the beginning of the gene. So it's a body of the gene plus 10 kb upstream and downstream. So where we consider those SNPs, and which means all other SNPs which are not in that area, they will be skipped. So actually we have to increase this area to let's increase it to 50,000 because we want to now consider also SNPs like that one which I showed, which in fact is not really in proximal region of a gene, and we don't know even does it actually changing that gene expression. It does actually, right? It was studied that in fact that SNP is in fact changing expression of that particular non-coding RNA, but it's still located pretty far from it, and that happens. You know, the enhancers can be very far from the start of transcription. So we put here the cut of 50,000, and then the region which we analyze around the gene, around the SNP, tells us actually it will answer the second question. Now it will answer about those motifs which are enriched in the region around the SNP. Not yet the motifs which are affected by the SNP itself, but the motifs which are enriched in the region. And that's, as I told you in the beginning, that's very important, but because the whole region is actually important, and the SNP is just a marker of that region. Okay, so and we can change the output, and then we simply start the analysis. And until it's working, what is important actually is to consider which set of motifs we are analyzing here. And here it's opening the result. In the meantime, you see it shows the progression, and it's called the profile. The profile is a set of motifs, set of positional weight matrices we are now analyzing. And the default here is a so-called non-redundant set of matrices. It's a pretty small set. As you may know, the TRANSFAC contains more than 7,000 positional weight matrices for vertebrates genome, but this non-redundant set contains several hundred only, so just one matrix for a group of matrices. This just to illustrate it, the analysis itself, so then it runs very quickly. But if for the real study, I would say you need the full set of matrices, and for that you simply click here, and you go here into the whole TRANSFAC profile section, and here you can select the bigger set. For instance, you can go to vertebrates, vertebrate, human, P001, which means these are all matrices, all positional weight matrices, which are for vertebrates, so which means all species which are belong to that big group of species, and then the cutoffs for those matrices are adjusted in such a way that it guarantees that each matrix gives approximately one hit to 1,000 nucleotides in the human genome. So that's why it's a human P001. So then you can select such profile. But as you see, we have started with a smaller profile, and our pipeline is almost ready. So we can already start looking at some results. For instance, it outputs such a diagram, which is simply shows in which genes these SNPs are found. And so there are genes where you see just one SNP. There are genes where many SNPs. And here these blue boxes are actually exons, and the SNP is shown by this red bar. And you see in most of the cases it's located in the intron areas, or or sometimes you see it in the 5', like in this gene, it's near the promoter, and so on. So you see such a diagram which gives you a sort of overview of where are its SNPs, and then yeah, it's still running. So we can actually go to the pre-computed analysis. I done it before starting our seminar. So it's here. So that's the output which you see. So what it does, it actually first split the SNPs according to where they are located. So whether they are fitting the exons of the genes. Okay, there are some now finished. So let me close that. All right. So they are located in exons, or they are located in regulatory regions. By the way, I don't know if you can see this. Maybe I have to increase the font a bit so you can see it better. So let me do it like that. So you see here that that's a folder with the results. These are SNPs in exons. These are SNPs in regulatory regions, which means regions either 5' regions of genome, 3' regions of genome, or introns. And then the other results are showing that SNPs. And then first, let's have a look at those SNPs which hit the exons. There are only 32 SNPs which actually fit the exons. Well, do they do anything bad or good to the proteins which are encoded by those genes? For that, we can see the result of the SIFT analysis. SIFT is a pretty good software which is integrated in our pipeline. You can also find it as separate tool, which checks each SNP if it's located in a coding region of a gene, whether it's changing a certain amino acid, and whether it's a very important for the protein structure or not. So you can see the prediction of the importance on the protein. In this case, as you can see, although all these SNPs are hitting the exons, they are not changing the codons. So I don't see anyone which would change any codon. Basically, it means they are, in all of those cases, these are exons which are not coding for the proteins, so non-coding exons. Okay, that's good to know. And again, as you can see, we know a lot, and we can tell a lot about those changes that can happen in the protein. But 700 SNPs related to that very important disorder, and none of them is changing anything in the protein. And saying that there are no mutations which are happening in certain proteins, famous ones, which are related to aggressive behavior, these are social types, but that's very rare cases, and that's a real mutation. So, and here we see those SNPs, single nucleotide polymorphism, which means we all may have them. So they are simply distributed in our population. and they have a certain influence on behavior. Okay, so their influence is really soft. It's in the regulatory regions of genes. It's changing motifs, and that's very difficult to analyze, and not very many tools can do that. Okay, so we are now understanding what's happening with SNPs in exons. Now let's have a look at SNPs in introns. First, actually, let's put those SNPs on a genome track. This is okay, just what happens. You know, we started from this table of the SNPs, just based on the SNP accession numbers. But the first thing what the pipeline is doing, it converts them into a VCF track. VCF track is another type of file which one can click, and it actually opens a genome browser, and it shows these SNPs in the real positions. And if you click on them, you see which nucleotide is a reference allele, which nucleotide is an alternative allele. And then you can see them in more context. For instance, you can put here in genome browser the genes, the gene names. So you can put this track with genes, for instance. And then okay, that's one thing, and we can zoom out, zoom in to see basically more information about the genes around. So let's jump to a nearby gene, for instance. So it's a pseudogene, or if we zoom out, we see the genes and the SNPs which are located near those genes. So the first thing we want to go back to that particular SNP of our interest, which is located near that one non-coding RNA. So for that, we need to know that accession number. Let me find it out. So let's go to the table. So we have this gene. Let's simply Google it, just to know a bit more about this gene. So sometimes Google is very important tool for you. No, that's not a good one. So now I want to ensemble just to get this accession number and to go here we are. Go to that genome browser, and let's put here this accession number, and we can go to that gene directly. Okay, so what do we see here? In fact, that's exactly the region of our interest. So we see that long non-coding RNA, so this link 0000951, and that's the SNP which has this very high p-value of association with that disease, with that disorder. So this is the SNP of our interest. So and now we want to see which motifs are found around that SNP. It's very easy, so that the program has calculated it for us already. So it is here. It's called, this file is called TFDS around regulatory SNP. So we just drag and drop it here. You see we got these sites now. So we can zoom in a bit more to see those sites in a better way. All right, so now you see those sites which are around that SNP, and in the area 500 nucleotides left and right from that SNP. So, and this gives us already a set of transcription factors which might be interesting for us, which might be involved here in the regulation of the genes involved in that particular disease. By the way, if I don't see any questions from you on the Q&A, just don't hesitate, drop some questions. I will see that it will pop up. So maybe just about something what I'm talking right now, something is not clear, I will come back to that right away and explain it in more detail. So it's not a lecture. I'm just talking because there is no questions. Just give me more questions, and I will stop talking and we'll start answering your questions. Oh, right. So I see the first question. So 2 questions. Is it possible to use TRANSFAC to calculate p-values for binding with a specific allele at a SNP and to rank predicted TFs based on their binding score? Yes. Right. And another question, can I get a free trial period for using your database? So for the second question, you should, yeah, there are various options, and you should contact our sales team. So they will talk to you about that. But about the first question, to calculate the p-values for the binding and to rank SNPs specific allele according to predicted TFDS. Yes, that's exactly what I was going to tell next. So now the next is really to understand cases when the SNP is actually changing the motif themselves. So for instance, in this case, you see there are now a number of motifs in that whole region, but there are motifs located just under the SNP. And that's the motifs found in the reference genome. For searching for the SNP, for the SNPs for the motifs in the region, we're using the reference genome, right? So, but SNP is changing the nucleotide in the reference genome, so it may affect those sites and those motifs. How to calculate that? And to do that, we use actually a different tool. It's called mutation effect on transcription factor binding. It's a very simple thing. So it has a different interface. And but the input of that tool is, which is important, is not just a list of SNPs, because they are RAS numbers, but we have to put here the track, already pre-computed track. Or if you have your track already before, you can upload it into the system directly as a VCF file, as I said, and we have several videos how to do that. You can have a look at our website for the videos. But now we have this track. It's here. This is our mutation. By the way, you see here we have 622 SNPs. And you remember that there were 700 something. So which means we lost around 80 SNPs, which probably were not in that areas around the genes of our consideration. So that's a part of that pipeline, and you can vary this flanking region to also grab other SNPs. But that's the life how it is. So now we take this track and drag and drop here, and actually as a profile, we want to use here a bigger profile, and there is a whole world of working with profiles, and that's another story. We were talking about that quite a lot before. Actually, what is a profile? Profile is a selection of motifs you want to use, and why not using all of them? You can use it, of course, and as I said, you can put here the whole set of motifs, but then your results might be quite bulky and very difficult to analyze. So you can make some focus studies. For instance, here I first took. I did it before. I took those motifs which I found significant in my region analysis. So actually looking at those transcription factor binding sites which are enriched in the whole region. And now I want to see which of them are also affected by the SNP itself. So for that, I prepared a special smaller profile. Where it is. Let me try to find it. Let's see. It was here. Okay. Right, right, this profile. So this profile, again, if I open it, it contains just 219 motifs from position weight matrices with their cutoffs, which are found significant in the regions around SNPs. You know, again, we started from 7,000 motifs. We ran the whole region. We found now 219, very small subset, but quite focused on only those motifs which are significant in the whole region. And now I want to understand which of those motifs are actually also changed by that particular SNP. So I take this profile and I put here in the default profile, and then one thing I have to put as a parameter is what is the delta of the score which I see significant. And in fact, there was a question here from Alexei that it would be interesting to calculate it in terms of p-values. This we can also do, but it's a little bit more complicated story. Right now, in this particular pipeline, I just talking about the score. And I can say I'm interested in the change of the score, which happens more than 0.3. You remember the score can be from 0 to 1. 1 is the biggest score, which means it's a maximum fit to the consensus. Around 4.5 is a random score. And we will observe those changes, which drops the score on the delta around 0.3. And that's according to our p-value calculations. It's around the p-value of drop of p-value over 1 order of magnitude in average. All right. Now we just run this. And now what it's taking, it's taking again the whole list of SNPs, of 600 SNPs, and it starts computing, taking each SNP individually and computing which effect this SNP happens on different motifs, whether it will kill the motif or will create the motif, and then it will list those motifs in a file. All found motifs will be listed in the file, and then you will see also you can rank them by this score and by the delta. All right, okay, it's computing right now. I have done it before. You can go directly to those results and put them on our genome browser. All right. So actually, these are the results which I see before. Right. These are the results. You see. We'll just wait until it's, because I did several, and we can just simply wait when it's finished. Finishes. Like this. All right, that's completed. Our previous part. By the way, you can indeed go through all the history of what we did. Now it's opening. Yes. So these are the first results. So what we see here is the site gain permutation. So this is a for instance, we can sort it by site gain or site loss. So you see the particular motif, for instance, FOX or 4 or pole to F, and here you see a frequency of how many of those mutations of those SNPs which we consider are actually changing. They lead to the gain of the sites or they lead to the loss of the site. And you see that, well, at least you can sort those motifs by the number of SNPs, or the frequency of SNPs, which are changing those motifs. And you see that by this sorting, you actually can see which of the motifs are most affected by those SNPs. We can have also, I will show very briefly how to put this estimation of p-value on that. But just now you can simply sort and see which of the motifs are actually most affected by this. You can also see them by them, them as basically here on the genome browser. It adds now these two tracks. So this track is a track of the losses. You see that's a loss, and this track is a track of the gains. And there is for this particular SNP we are analyzing. In fact, there is no site which was gained, but there is one site which is lost, and that's this CTCF site. We can have a look at what is the delta of that loss, and the delta is pretty big. It's actually minus 0.075. It's practically kills the site, we can also see what happens. Actually, you see that changes reference G into the A, and which I can tell you right away. Basically, it's in fact changing that. So if you look at the consensus, let me you see this site is going in the minus strand from right to left. So which means this consensus has to be converted. In fact, this change of G into A is changing this big C into T, which completely destroys the whole site. So it means this in the reference genome, this is a very good site for CTCF. And the alternative genome, which is actually correlated to that particular disorder, this SNP destroys that site. And also you see these CTCF sites are very important in general. They are overrepresented in those regions of our interest. And by the way, CTCF is a very important factor. It's actually a factor of an organization of a chromatin, and that's actually so-called insulators. If it's not happening, if a factor cannot bind to that place any longer, it's a very big change in the structure of that chromatin. That's why they are so important. And that's the one example how we can analyze the SNPs and create hypotheses about importance of particular transcription factors in that SNPs. So now, very briefly about the p-values. So there is a tool here which is sort of secret tool. It's not among these tools in the starting page. To find it, you have to go to the analysis method to individual methods and go here to NGS analysis, because this tool is in fact mainly used in studies of the VCF files. When you have a lot of SNPs of interest, not just 700, where you can make really a good statistics. But that tool is called mutation effect on sites. And here, the mutation effect of sites is the same. Mutation effect. Sorry, that one is the one I was showing to you. That was another one. Site yes, compare TRBS mutations, or it's this one. So this tool is located in the site analysis section of our platform. What it does is actually doing complete statistical analysis. So you put here your VCF file, which again, the same as before, you put it here, but then you need the background, as I showed you in my lecture, in my starting presentation. So you need a background, and for that we need a random set of SNPs. And for instance, here we have in our public section, in the supporting data, we have a random set of SNPs. We call it actually a random VCF, which has a 10,000 position, random position in genome, and where we randomly change the nucleotide into random nucleotide. So it's kind of very random background. And then now it has what to compare it with, and then it will start to calculate, to find binding sites for each SNP, and then also finding binding site in these random SNPs, and then it compare the frequency of found binding site. And then it takes some time in doing this comparison, but let me show just the final result of that p-value calculation. For that, I will see I did it. Here. Okay, so I will I will show it in a bigger context, but that's another story, which I will spend maybe next time. We have actually a tool called Genome Enhancer, which can do everything what I was telling to you automatically. So don't spend time on doing all this by step. You just load this SNP list and it does everything. It does calculating SNP frequency in the whole region. It will calculate sites in each particular position. And it also will calculate the statistics of changing or gaining or losses of sites. And it will generate a report about that. And it has many other things in that report. And I will just show you one part of that report, which concerns those statistics. So you see, it actually tells you a lot about those SNPs, about what these genes are doing and to which disease they belong to. And then it creates this table of statistics where you see these p-values. Yeah, we need p-values, right? So we can open, well, we just can directly look at this table right away. So it tells you actually list those SNPs, which are, list those motifs, which might be either destroyed by the SNPs or created. And then it counts, calculates how many cases it observed those changes. Like, for instance, it observed that in 5 cases, in 5 SNPs, it leads to the gain of that site, of that motif. But in the random background, it happens only in 3 cases. And of course, you know, 5 times out of 700 is more significant than is significant difference from 3 times out of 10,000, right? So it's more frequent case. Although the statistics is pretty poor there, it's just 700 SNPs. But still, you see some of the motifs. For instance, this motif actually losses reaches, well, I would say this ATF2 is a bit better. So losses reaches in the 10⁻⁴, which is in the region of really good statistics. But as again, on a SNP data, when we are talking about just 700 SNPs, this statistics is not really, you know, it's not big enough to tell and to lead to really conclusive statistical conclusions. So much, sorry, much better this when you have a much bigger number of SNP of changes. For instance, if you are analyzing the whole genome and you're analyzing not just SNPs but variability of genomes, and you are talking about you know, 10,000 changes, 100,000 changes in the whole genome. For instance, we have here VCF file in the case of non-small cell lung carcinoma. And in that case, those statistics I can show to you are much more convincing. So let me go to that particular type of table. You see there are much more changes you observed in when you sequence the whole genome, not just checking individual SNPs here and there. Yes, here is the table. And you see here, for instance, there are much more significant, like 10 minus 10, 10 minus 16, for instance, which tells you with a more statistical power that indeed these motifs is statistically significantly affected by a number of changes in genome. And the reason is that because, well, in case of cancer, that may be a result of a selection, which select the whole genome from a particular presence or absence of number of motifs in the whole genome. In case of SNPs, that also may be a certain drive when you correlate it to you found a certain SNP correlating to a certain disorder, right? So that leads to these statistics as well. All right, so that's what I was going to talk today about those SNPs and motifs. So that's time now to write some of your questions. I have some more time, so if you have time, just write questions. I can briefly answer them. So also, it's time for you, if you are actually watching this video, not online, but in a video or in YouTube, because we will put it in YouTube. So your guys in future, please put your like under this video, because you like likes. So that's time for you to put the like. Okay, so if you don't have any questions, then thank you for your attention. Thank you very much, and see you next time.